Wrapping up our lab on the standard plate count and the serial dilution. So hopefully you got a chance to look at the video clips that were explaining about serial dilutions. You would have done it slightly different from what was shown in the lab. We have the 99 milliliter bottle, so you would have done a 1 to 100 dilution each time. So 1 to 100 on bottle A, 1 to 10,000 on bottle B, and 1 to 1 million on bottle E, and then plate it out exactly as shown on the 4 plates that you can see right here. So you would have done exactly these 4 plates, either plating 0.1 milliliter or 1 milliliter out of bottle B, or plating 0.1 milliliter and 1 milliliter out of bottle E. You would have gotten plates that some of them would have been too hard to count because too many colonies, and some of them would have had so few colonies on it that the result is also inaccurate. In addition, you would have done the optical density determination, and then you should have gotten, the goal was to establish a standard curve that correlates optical density with actual colony forming units. In other words, we can use the optical density measurement together with our plate count results to establish a standard curve that gives you then an idea by just measuring optical density, how many colony forming units do I have in my culture. This should be done for every type of bacterium separately, but once you have a standard curve like that, you can use it and then simply measure optical density. And that was shown in the second video that was posted on Canvas. So now here this is actually the spectrophotometer we have in the lab. You would have gotten to use it, measured optical density, and then should have gotten some results. For the optical density measurement, you should have just diluted the bacteria. So undiluted 1 to 1, that's this one right here. This one usually gives an optical density of over 1, which is also inaccurate. It's just too thick. The optical density is just there's not much light coming through. Then 1.1 to 2 dilution, that should have gotten you a reading that's usable. 1 to 4, 1 to 8, and 1 to 16 dilution should have given you some reasonable results. And so here I put some sample data here out there for you that are pretty reasonable. So the 1 to 10,000 dilution should have gotten you probably too many colonies to count. If you have a plate that has a lot of colonies on it, sometimes what you can do is so you take your plate and you put it in quarters, and then you just count on one side here on the quarter, count these guys, and then you multiply times 4. That's what you typically do, and then you get an estimation of the number of colonies. So I just estimated here for 2,000 colonies. Then the 1 to 10,000 where you only plated 0.1 milliliter, so that gives you another 1 to 10 dilution. So this one, the dilution is 10 to the 5th. So because you've plated 1/10 of what you plated here out of the same bottle, of course your dilution is a factor of 10 higher. So we go from 10 to the 4 to 10 to the 5 on the dilution factor. And that, let's say, as an example, gave you 320 colonies. That's relatively easy to count. But again, if you don't want to count all 320, you can still use the method up here where you are splitting the plate in quarters and then just count 1 quarter and then multiply times 4. On bottle C, if you plated 1 milliliter, then that would account for a 1 to 1 million dilution, and the dilution factor then 10 to the 6th, of course, and that, let's say, gave you 35 colonies. That's really easy to count. And then the last one here, 1/10 of a milliliter of this dilution, the same dilution, but only 1/10 of the volume plated, that gives you a dilution factor of 10 to the 7, or 1 to 10 million. Now, how do we go back to the actual colony forming units in the original culture? Well, you have to multiply the number of colonies that you counted times the dilution factor. So that means you will take the 35, let's say we're starting here with the 35 colonies right here that we counted on our 10 to the 6 dilution factor, which was 1 milliliter of the 1 to 1 million dilution. So that was bottle C. You take 35 times 10 to the 6, and most people don't want double digits here on the before they express an exponent. So in the scientific notation, you usually go then 3.5 times 10 to the 7. Or here, let's say, calculate our result on this plate. It would have been 320 colonies times the dilution factor of 10 to the 5th. So that would be 3.2. So I'm going to take 2, move the decimal point 2 places over, and put these 2 decimal places into the exponent. So now I have 3.2 times 10 to the 7. Pretty close. As a general rule of thumb, you want to count plates that have somewhere between 20 to 500 colonies on them. So these 2 would be the ones that you would want to count. Of course, it makes your life easy if you just count 3 guys, but that usually has a high level of inaccuracy built in, of course. So be sure that you know how to calculate the number of CFUs in an original culture when you're given a dilution factor. It's real simple, but as always, you got to know how to do it, and then everything is easy. So you take the number of colonies that you counted times the dilution factor, and then you just express it in scientific notation, because most people don't like to write all the zeros out. Now, let's move on to the optical density determination. I also gave you some sample data that I just made up. And these optical density data, they were supposed to be plotted. And of course, if you were diluting 1 to 1 to 1 to 2, then you should get approximately half. One thing to point out is that an original culture has such high optical density that this reading will be inaccurate. So this will be outside of the range of your instrument, basically, and it just gives you some sort of a nonsense reading. It just gives you the highest reading it can read, and that's pretty meaningless usually. So you want to be below 1 to have meaningful data. And so on our 1 to 2 dilution, that's where we are reaching that level where we are reasonably satisfied with the accuracy of our instrument. And from there on, it should be roughly half. So I just made up some data that reflect that. And the 1 to 16 dilution, we get an optical density of 0.1. Then at 1 to 8 would be 0.2, ideally. And then here it should be 0.4. Let's say we measure 0.45. And then here it should be then doubling again, of course, that will be 0.9, but it's 0.85. So just gave you some data that are reasonably good. And from there, you put those data into this plot right here on this worksheet. And then you were supposed to put a regression line through that, so a best fit curve that accommodates most of the points, so that you would want to make try to be a linear curve. Try to ignore this point right here, because this one is above 1, and we consider optical densities above 1 to be not reliable data points. So we want to make a nice straight line through here. So could look something like this. That's pretty good. If you're doing this on a spreadsheet program like Excel, then you can ask Excel to give you the correlation coefficient and give you the equation of the line that you have right there. All of that is useful. I didn't put this data into Excel, but that normally would be done. But this curve here, or this plot, isn't really that useful. Remember, you would want to have a correlation between the number of bacteria and the optical density. Now, these are on the x-axis. These are dilution factors. That doesn't really tell you anything. You want to know how many bacteria were in there, and so for that you need the data from the optical density determination. You need to correlate that with the plate count data, so up here. And for that, ideally you would want to get a plot that looks like this. That's the one that correlates the optical density now to actual numbers of bacteria, live bacteria, colony-forming units, those bacteria that were alive, settled down on agar and formed a colony. Once you have such a standard curve, you can use it over and over again, just measuring optical density of your bacterial culture, and you should get, you would have an idea of how many live colony-forming units you have in your test tube or in your flask. Now let's answer the remaining questions here. What is the maximum optical density that is within the linear portion of the curve? You don't want to have any kind of readings above 1.0. So that's just the rule of thumb. On question D here, what is the corrected or true optical density for the undiluted culture? So remember, at this point we consider this one here inaccurate because our instrument just can't measure the true optical density. What we're going to do is we're going to take the 1 to 2 dilution and double that value. So the true optical density of the undiluted would be 0.85 times 2, and that would be 1.7. So this actually should have been 1.7, but our instrument is not able to pick that up. So that's the answer to D. What's the correlation between the optical density and the cell number for your culture? Of course, as the cell numbers go higher, the optical density goes higher, and you would have this correlation right here. And again, if you use Excel, then you can get the equation for your line and the correlation coefficient, which hopefully would be near 1. That would be ideal. Going down here, the short answer questions. When performing a standard plate count, why are the counts reported as colony forming units rather than cells? So you want to distinguish between those cells that are able to form a colony versus a cell that might be near death or is already dead. You don't know. A cell may or may not be able to form a colony because it's already old or dying or already dead, and then it cannot form a colony anymore. So you really only want to count live cells that are able to form a colony. How would you inoculate a plate to get a 1 to 10 dilution or 1 to 100 dilution? Starting with the 1 to 10 dilution, so what you would do is you would take 1 milliliter of your original culture, put it into 9 mLs of water or LB broth medium, and then take 1 milliliter of the 1 to 10 diluted medium and plate that onto an agar plate. For the 1 to 100, you can either use the 1 to 10 dilution and do another 1 to 10 dilution, or you can go back to the original culture. For 1 to 100, you could put 1 milliliter of your original culture into 99 milliliters of water or culture medium, and then take 1 ml of that diluted medium and plate it onto a agar plate, nutrient agar or something like that, and then count the colonies that you're getting after incubation. Now on 3, why is it necessary to perform a plate count in conjunction with the turbidimetry procedure? Well, as we said before, turbidimetry will give you only particles. Whether they're dead or alive, you don't know. Just like we talked about in question 1, the answers to that. So we really need to find out how many colony-forming units we have, live bacteria, and correlate that to an optical density, and that's why we need to do both. On 4, the last question, for the following methods of bacterial enumeration, does the method determine the total count or viable count? Starting with the MPN, the most probable number, that one will give you an idea of the viable count. It will give you a range of probable live bacteria, probable number of live bacteria in your sample, and this is usually done in the water district or places that want to have an idea of how many live bacteria they have in a certain sample. Now the microscopic count, that one will give you just a total count. So you don't know what you're looking at, whether it's alive or dead. You have no idea. You're just looking at a bacterium. Sometimes you can tell if they move, and you may not have a moving bacterium, and then you just don't know. The standard plate count, of course, gives you viable colony forming units, and the turbidity measurement, that one determines total count, as we said before.